Hi, it's me again, Andreas, but this time I don't talk to you in my function as the chair of the Cost Action EU-ROS, but as a member of it. Yeah? I'm of course also a workgroup member, and I have special interests in the redox biology and oxidative stress field, and I would just like to briefly mention our main interests at the moment in the lab, and yeah, give you a brief overview on the research we are doing regarding reactive oxygen species. I'm originally coming from the biochemical research field, and I worked several years with Volker Ulrich in Konstanz, and we were mainly interested in targets of peroxynitride, especially in metal catalysis of nitration reactions. So we worked a lot on P450 enzymes and how they catalyze nitration of nearby tyrosine residues. Prostacyclin synthase nitration was one of the big topics in our lab. Then we were also interested, of course, in nitrosation reactions because they are reversible. It's of large interest for the redox biology community. Then I moved from Konstanz to Hamburg to the group of Thomas Münzel, and I rather engaged then in preclinical research, working in animal models that are related to cardiovascular disease and oxidative stress. So therefore we had very nice models in Hamburg on atherosclerosis, hyperlipidemia, hypertension, hyperglycemia, of course. And finally, of course, one of the big topics in our lab, and I'm proud to say that we are probably a leading lab on nitrate tolerance in the world. So therefore probably some of you know that organic nitrates such as nitroglycerin under chronic use will induce some adverse side effects, such as induction of reactive oxygen and nitrogen species formation, as well as induction of nitrate tolerance, so the loss of the vasodilatory potency of the organic nitrate, but also cross tolerance, which is impairment of endothelial function. So therefore organic nitrate therapy can impair the endogenous relaxation pathway via NO and cGMP. So this was one of the big topics we worked on, especially since the new organic nitrate bioactivating enzyme, the mitochondrial aldehyde dehydrogenase, was discovered by the research group of Stemler and co-workers in 2002. We were also very much engaged in this protein because it's highly redox regulated, it has several thiol groups in the active site, and therefore is easily inactivated by oxidative stress. Then other big topics in more recent years now is interaction of oxidative stress with inflammatory pathways. For example, we are very much interested in the role of inflammatory cells in development of hypertension, and I think this is an emerging field at the moment that also large companies have discovered that anti-inflammatory therapy in cardiovascular disease is one of the future, I would say, targets of interest. So therefore we are interested in all kind of interaction between oxidative stress and inflammatory pathways. We are interested in antioxidant and anti-inflammatory properties of all kind of drugs, and recently we have discovered quite nice properties of DPP4 inhibitors and GLP-1 analogues in order to suppress inflammatory pathways and oxidative stress in sepsis models. So therefore this was quite unexpected effects of DPP-4 inhibitors and GLP-1 analogues that were initially developed as anti-hypoglycemic drugs in order to decrease high blood glucose levels. So therefore that they really have quite potent anti-inflammatory and antioxidant effects was quite surprising. Okay, then in addition we are also working, of course, on some molecular mechanisms. There my major favorite topic at the moment is ROS-induced ROS. So therefore I'm interested in all kind of pathways how reactive oxygen and nitrogen species oxidative stress can induce further production or formation of reactive oxygen and nitrogen species by other sources, by secondary sources. So therefore this is the kindling or kindling radical concept, where you have initial formation of some reactive oxygen and nitrogen species that are able to trigger further activation of other sources, and thereby of course further increase and amplify the initial ROS formation. Okay, so therefore I think this is the major interest in redox biology and oxidative stress research in our lab, and I hope that also the other members just show you a bit what they are working on. Bye.